live. Yeah. We have started.
Looks like it is gone live.
Sir.
So shall we start?
Just 1 minute because this is 8:59. Yes, Abha, you can start now.
Okay, thank you, sir. With warm greetings, a very good morning to everyone. On behalf of CSIR-CIMAP, I, Dr. Abha Meena, extend my warmest welcome to Honorable Dr. Shekhar Mande, DG CSIR, Dr. Prabodh Kumar Trivedi, international national speakers, dignitaries from other institutes, my colleagues and the of inaugural program of International Virtual Conference on Plant Science Specialized SMME 2020, organized by CSIR-CIMAP. To begin with, the lighting ceremony as a tribute to Mother Saraswati, the goddess of knowledge, art, and to make this event a blessed one, we invoke Goddess Saraswati by playing one lamp of knowledge and wisdom.
तुषार हार नवना या शुभ्र वस्त्रामृता या वीणावरदण्ड मंडिता या श्वेत पद्मासना या ब्रह्माच्युतं शङ्करं प्रभृतिभिः देवैः सदा वन्दिता सा मां पातु सरस्वती भगवती
again welcome to all. Moving forward, let me give us some information about CSIR-CIMAP. Central Institute of Medicinal and Aromatic Plant, popularly known as CIMAP, is a frontier plant research laboratory of CSIR, established as Central Medicinal Plant Organization (CIMPO) in 1959. CIMAP is steering multidisciplinary high-quality research in biological and chemical sciences and extending technologies and services to the farmers and entrepreneurs of medicinal and aromatic plants. CIMAP contribution has made India the global leader in mints and related industrial products. Research work on anti-malarial plants, Artemisia, is another revolution catalyzed by CIMAP, which brought CIMAP at the frontier globally. CIMAP has been designated by PPV and FRA, Protection of Plant Varieties and Farmers' Right Authority, as nodal laboratory for plant varieties protection and DUS, distinctiveness, uniformity and stability, testing of medicinal and aromatic plants and seed species. CSIR Aroma Mission and CIMAP Biomillage missions are expanding the industrial cultivation of appropriate aromatic and medicinal plant across India, empowering Indian farmers and aroma trade. International Center for Science and High Technology, United Nations Industrial Development Organization ICS-UNIDO has recognized CSIR-CIMAP as focal point for Southeast Asia. CSIR-CIMAP is also a nodal center for ocean, that is IURA RCST in end. CSIR-CIMAP scientists established the whole genome sequence of Ocimum sanctum, which is commonly known as holy basil. Thank you. I would now request Dr. Dinesh Nagegoda, Senior Principal Scientist and the convener, to give his opening remarks and brief about the conference. Dr. Dinesh, please.
Thank you, Dr. Abba. A very good morning, good afternoon, and good evening to all who have joined us from India and different parts of the world. On behalf of the Director CSIR-CIMAP and the organizing team, I extend a warm and hearty welcome to all of you to the International Virtual Conference on Plant Specialized Metabolism and Metabolic Engineering, which is abbreviated as PSMME 2020. As chief guest of the inaugural function, we are really honored and privileged to have with us Dr. Shekhar C. Mande, Director General of Council of Scientific and Industrial Research and Secretary, Department of Scientific and Industrial Research, Government of India. Thank you very much, sir, for taking your time to grace this occasion. We also have with us Professor Natalia Dudareva, a distinguished professor of biochemistry from Purdue University, USA. Thank you, Professor Dudareva, for joining this session and agreeing to deliver the inaugural talk. To tell you all briefly how this event came about, since physical conference was not possible due to COVID-19 pandemic, we conceptualized this virtual event to bring together the scientific community so as to discuss the latest happenings in various aspects of plant secondary metabolism and engineering. We planned this event sometime in July 2020 to be conducted in October 2020. It was initially planned as a two-day event. However, since we received an overwhelming response for our request from the speakers, we were happy and excited to extend it for 3 days. We equally got a huge and enthusiastic response from students and faculty for participating in this event. In this conference, we have 35 eminent invited speakers. Among them, 21 are from India, 3 from USA, 2 speakers each from Germany and Singapore, 1 speaker each from Canada, France, Australia, New Zealand, Israel, and Hong Kong. On behalf of the organizing team, I would like to thank and extend a warm welcome to all the esteemed invited speakers for readily accepting our invitation to deliver a talk at this event. Besides invited speakers, the conference is being attended by more than 450 registered participants, which includes 70 submitted abstracts. Among them, 17 have been selected for oral talks to encourage young researchers. Complementary to this live event, we also have included a poster session being conducted via conference Twitter account, through which the participants can display their posters. For this, we have provided a 4-slide poster template for optimum display in the Twitter. As an encouragement to young researchers, we will be awarding the best selected oral talks and posters at the end of the event. So this conference is unique in the respect that it is the first such conference to be conducted virtually with such a large gathering of scientific minds from across the globe in this specialty theme area of plant secondary metabolism and its engineering. We have tried our best to put together a great mix of researchers as speakers from different parts of the world that includes senior and eminent researchers who are a kind of authority in their field and some mid-stage and young researchers in different aspects of plant specialized metabolism. For the next 3 days, we all can look forward to a feast of scientific talks on 6 different themes ranging from how secondary metabolites are formed in the plants, their role in plant growth and development and ecology, how the formation of specialized metabolites is regulated, and how they can be overproduced by engineering. And finally, the conference will cover the application of these plant secondary metabolites in human health. As a convener of this event, I take this opportunity to extend sincere thanks to my director, Dr. Prabodh Trivedi, who provided a great support in organizing this event. Also, I thank and appreciate the efforts put in by the organizing team both in Lucknow and in Bangalore in making this event happen. Finally, on behalf of the organizing team, I once again welcome all of you to this event and wish you a great conference ahead for next 3 days. Thank you one and all.
Thank you, Dr. Dinesh, for the brief. Now I request Dr. Prabodh Kumar Trivedi, Director CIMAP, to give his welcome and the chief guest, Dr. Shekhar Mande, DG CSIR. Sir, please.
A very good morning from India. We are delighted to have you all amongst us through this virtual platform for the inaugural session of the International Conference of Plant Specialized Metabolism and Metabolic Engineering 2020. On behalf of the CSIR-CIMAP and organizing team members, I extend warmest welcome to all those connected in this session or will join us during the conference. It is difficult for some of the delegates as they are from various countries and time zones are different to join at this moment. However, all of you managed to join. Those who join us, as we cannot meet physically due to travel restrictions and social distancing due to COVID-19. This shows that scientific community understands each other very well and is ready to go to extra miles whenever required through different modes. Today we have a person with us as the chief guest who has always inspired us and need no introduction. However, in the interest of younger colleagues and delegates, I will provide a glimpse of his scientific journey and leadership. Our chief guest is Professor Shekhar C. Mande, the Director General of Council of Scientific and Industrial Research, which is CSIR, as well as Secretary, Department of Scientific and Industrial Research, DSIR, Government of India. Professor Mande is a well-known structural and computational biologist. His research interests have been structural biology and X-ray crystallography, structural characterization of Mycobacterium tuberculosis proteins, and computational analysis of genome-wide protein-protein interaction. So he has done tremendous work in these areas and is leader in all these areas through his path-breaking research. Professor Mande did his master's in physics from University of Nagpur and PhD in molecular biophysics from the Indian Institute of Science. Following his PhD, he joined as postdoctoral fellow in Groningen University, which is in the Netherlands. After working in University of Washington, Professor Mande returned to India in 1995 and joined CSIR IMTech, which is part of CSIR, to serve the country. In 2001, he joined senior staff scientist at Center for DNA Fingerprinting and Diagnostics, which is CDFD. In 2011, Professor Mande became the director of the National Center for Cell Sciences, which is in Pune, in India. And after that, in 2018, he is leading CSIR as the Director General. This organization has about 40 laboratories all over India, and he has shown that how to connect each lab together for the cause of this society as well as for the nation. Professor Mande has been conferred with several awards and honors. To name a few, his B.M. Birla Young Scientist Award, which was conferred in 1999. He was awarded Sant Sarup Bhatnagar Prize for Science and Technology for 2005, which is the highest science award in India in the biological science category. He is fellow of all the major academy like Indian National Science Academy, New Delhi, National Academy of Sciences, Allahabad, and Indian Academy of Sciences, Bangalore. More importantly, he has led the entire CSIR team, which has close to 40 laboratories across the country and the largest scientific institution to work for the society and in the country. During this difficult time of COVID-19, he has shown tremendous leadership. He encouraged all of us to work for society and come out with solutions through in-depth science. There are several and diverse success stories which have emerged due to your leadership, sir, and are making the country proud. Thank you, sir, for giving us time and inaugurating this conference. Thank you, sir.
Thank you, Prabodh. Prabodh Trivedi, my senior colleague, Professor Dodareva, all the people, all family CSIR-CIMAP family members, everyone who has joined across the globe in this conference. It's a great pleasure for me to be able to be with you today on this particular event. All the people around the world who have joined the conference may know that the Central Institute of Medicinal and Aromatic Plants, what we very favorably call as CIMAP, Lucknow, has been one of the leading institutes in the country in promoting medicinal and aromatic plant cultures. Some of you might know that India did not have much mentha production about 60-70 years ago, and today India is one of the largest producers of mentha, and it is thanks because of the efforts that have been put in by institutes such as CIMAP Lucknow, but also very ably assisted by some of the other CSIR laboratories in the country. As Prabodh quite clearly told you, that CSIR has a chain of about 38 laboratories across the country, and we work on many different varied areas such as aerospace, mining, petroleum, and so on and so forth. And in the plant kingdom, we have at least 4 or 5 different institutes which are completely devoted to working on different aspects of plant kingdom. We are very proud of them, kind of work that they have done. In the past few years, particularly CIMAP Lucknow has very ably led a program on what we call as Aroma Mission, in which many farmers in different corners of the country are being taught how to cultivate aromatic plants and how to extract value from these aromatic plants. So that program we are very proud that CIMAP has led very successfully. It's a very visible program in the country, and all our accolades to CIMAP for having led that particular program. The very fact that we are meeting today online only is a reflection of the times that we are living in. About 9 months ago, if anyone had told us that this kind of time is upon us, I think most of us would have dismissed this as science fiction. It's in no time we realize that indeed something unusual has happened. The unusual has been predicted by scientists all over the world for several years, but has not been reconciled by the fact and only showed that humanity was not prepared to this scale of the problem that would be upon us. And in fact, when I talk to popular media, newspapers, TV channels and all, I have been very vocal about this fact that scientists tell you something very, very useful, and larger society and the policymakers and politicians must listen to scientists what we have to say. We have been telling you that the next pandemic is around the corner and we are not prepared for it. And indeed, today what we have is that situation. We have been telling you that climate change is real. We are not prepared for it. There's the kind of things scientists have been telling, and these are very informed technical opinions. And our society, our policymakers, and everyone else must reconcile to the fact that these opinions are based on facts. And the opinions are not imagined fantasies of some nerdy people, right? And that's exactly what people should learn to respect our opinions on this. In some respect, the corona pandemic has brought the world even closer. And that's a reflection of the fact that what we see here, that so many people have gathered here to also listen to Professor Dodoreva very soon, that people have actually gathered online and have been discussing science with a proper depth as it is. In fact, it reflects the resilience of the entire scientific community around the world, how it has risen to the challenge of the pandemic, and in the time of the pandemics, that how we can get together and work together. Now, I am not a plant person myself, as Prabodh said. Most of my work has been on bacterial kingdom, or how bacteria affect humans. And favorite model system in my lab is in Mycobacterium tuberculosis. But I have had chance to work on metabolism in the past few years. The metabolism within Mycobacterium tuberculosis, or the host metabolism when Mycobacterium tuberculosis infects the host, on both these sides. And indeed, I've been writing in my papers that the first change that happens in any organism due to a change in environment, whether it be a stress condition or anything else, is the change in the levels or the change in the concentrations of the metabolites, right? Gene expression follows this particular step, and expression at the protein level even follows further down. So the first thing that happens is essentially changes in the concentration of metabolites, and this is essentially mediated by all the secondary metabolites or secondary signaling that what we have. Now, plants being very fascinating and so complex, that is very worthwhile looking at these aspects, which all of you have been doing that. All the experts who are gathered here, all of you have been doing that, and I have enormous respect for all of you for trying to understand things which are so complex, apparently so complex, and trying to gain understanding of how plants deal with these changes in secondary metabolites, in changes in the concentration of the metabolites, and how that eventually leads to the response of the plants to the change in environment. I wish you all the very best. All the people who have gathered around the world, I'm sure, will feel enriched by these discussions. I wish all of you a very safe and healthy time wherever you are, and wish the next 3 days to be very fruitful and productive. Thank you all very much.
Thank you, sir, very much for encouraging words. We'll try to organize things in very proper way, and I hope that conference will go very well. Now, I think Dr. Dinesh will introduce the speaker for today.
Thank you, sir. It is my honor. Am I live? Okay. It is my honor and great pleasure to introduce the inaugural speaker of PSMME 2020, Professor Natalia Dudareva, with whom myself and Dr. Ajit Shashni, Chief Scientist of CSIR-CIMAP, had the privilege of working as postdoctoral scientists. Professor Dudareva is presently a distinguished professor in the Department of Biochemistry and an interim director of Center for Plant Biology at Purdue University, USA. She received her MS in Biology and Biochemistry from Novosibirsk State University, Russia, and her PhD training from two different places, one at the Institute of Biochemistry, Kiev, Ukraine, and the University of Louis Pasteur, Strasbourg, France. After a postdoctoral training at the University of Michigan, Dr. Dudareva joined the Department of Horticulture and Landscape Architecture, Purdue University, in the year 1997 and received a distinguished professorship in 2010. She joined the Department of Biochemistry in the year 2013. Research in Dudareva laboratory focuses on understanding biochemical and molecular mechanisms controlling the formation of primary and secondary metabolites, which include phenylpropanoids and terpenoids, in plants using the power of genetic and biochemical approaches combined with metabolic flux analysis and modeling. With initial focus on plant volatiles, her group has not only identified many genes involved in the final steps of their biosynthesis, but also discovered the primary metabolic networks that supply precursors for volatile formation in plants. Her group has also showed that active biological mechanisms are involved in transporting volatile compounds from plant cells to the atmosphere. Dr. Dudareva has published almost 130 papers, 28 book chapters and 3 books, and has given more than 200 invited lectures at the conferences and other universities. She has received recognition for her research as Purdue University Faculty Scholar and the Wickersham Chair of Excellence in Agricultural Research. She was awarded the Purdue University Agricultural Research Award, Sigma C Faculty Research Award, the Alexander von Humboldt Research Award from Germany, and in the year 2018, she received Herbert Newby McCoy Award, which is Purdue's most prestigious award given for outstanding work in the natural sciences. In the year 2010, Professor Dudareva was selected as a fellow of the American Association for the Advancement of Science, which is a prestigious fellowship. Some of the recent work from her lab has appeared in prestigious journals like Science, Nature Chemical Ecology, Nature Communications, and Nature Plants. With this brief introduction, I now invite Professor Dudareva to deliver our inaugural talk on plant volatile emission beyond the traditional view.
Thank you very much, Denish, for your kind introduction. Thank you very much for inviting me. It's a big honor to give a lecture. And you have amazing program for your conference, and I wish you all the best during all these 3 days. And today we are working in the world of volatile compounds. Could you see? Yeah, volatile compounds. And if you look around, we live in the world of volatile compounds, which are widely used right now in perfume, cosmetic and fragrance industries, which are constantly looking for new unusual scent. And biologically active volatile compounds and essential oils they're currently used in aromatherapy. And if we look on this diverse, amazing range of compounds, they are mainly produced by plants. And plants, they use these volatile compounds for their communication and interaction with surrounding environment. And volatiles are released from flowers, fruits, and leaves into the atmosphere, and from roots to the soil, they contribute to plant, animal, and plant-insect interactions. Volatiles emitted from flowers, they attract pollinators that ensure reproductive and evolutionary success of plants. In nature, structure of the flower, color, and scent are the main cues attracting pollinators to flower. While flowers could be exactly the same in flower shape and color, there are no two scents that are identical, exactly the same. due to wide variety of volatile compounds and their relative abundances and interactions within the flower bouquet. And successful pollination is not only important for plants, but also for human nutrition, since 1/3 of our diet directly or indirectly depends on successful pollination, including fruit trees, nuts, oilseed plants, and vegetables. Volatiles emitted from fruits, they contribute to taste and aroma of fruits and also attract seed disseminators. Volatiles released from vegetative tissue, they contribute to plant defense. They can directly repel or intoxicate attacking herbivores, or they can attract natural enemies of attacking herbivores, thus protecting plants via tritrophic interaction. And emitting plants not only reduce amount of attacking herbivores more than by 90%, but also can inform neighboring plants about pathogen attack. Release from view to below defense, also acting as they can repel or intoxicate root attacking herbivores, or they can protect roots via tritrophic interactions. Volatile compounds also contribute to stress protection and can be used as a pest management. Volatiles are also important for atmospheric, play important role in atmospheric chemistry, because they form these secondary organic aerosols, which serve as a nuclei for cloud formation. And if you will look on total emission of VOCs per year, it's 10 to the 9th ton of carbon released per year by plants. But why it's important to study volatile compounds? First of all, because we can increase yield of insect-pollinated plants. We can also study the role of individual volatiles in pollination attraction and plant-plant interactions. We can enhance the natural defenses of crop plants and provide an alternative pest management strategy that is based on biological control. We can also improve scent and aroma quality of flowers and fruits. We can, in such way, we can increase value of ornamentals, produce novel aromas for perfume industry. We can use plants as factories for new natural flavors for food industry and for biologically active volatile compounds for aromatherapy. We can also improve production of advanced biofuels and prevent unwanted effects of volatiles on formation of secondary organic aerosols and thus on atmospheric chemistry and climate. If you look on emission of volatile compounds, emission depends on rate of biosynthesis and rate of release. And rate of biosynthesis is species-specific, spatially, developmentally, and diurnally regulated. And even closely related species, which rely on different pollinators, and here I have example of Clarkia cancina and Clarkia borai, which rely on different pollinators, bees and moths, respectively. They produce different spectrum of volatile compounds. And during development, the highest level of volatile compounds produced by flowers when flowers are ready for pollination. And very often, emission is diurnally regulated and coincides with activity of potential pollinators. For example, snapdragon flowers produce volatiles during the day as they pollinated by bees, which are active during the day, and petunia flowers produce volatile compounds during night because they are pollinated by moths. In addition, rate of biosynthesis depends on physiological factors such as pollination in floral tissue and pathogen infection in vegetative tissue. And after pollination, floral scent does not go right away down, but it's practically flowers wait till fertilization takes place, and in case of snapdragon flowers, drop in floral scent occurs after 2 days, after 48 hours, because fertilization takes 36 hours, 48 hours here. And in petunia, it takes 36 hours for pollen movement and fertilize the egg. This is when the scent drops and decrease. If we look on rate of release, for volatiles to be released, they need to cross plasma membrane, cell wall, cuticle, and then they will release in the atmosphere. In this situation, rate of release depends on physicochemical parameters of compound itself, on volatility, diffusivity, and octanol water partition coefficient, and on physicochemical parameters of barrier which compound crosses, including membrane lipid composition, cell wall porosity, and cuticle composition and permeability. And release also depends on environmental factors, including light intensity, relative humidity. And the big question for us was whether biological processes such as sequestration, transport, and shuttling mechanisms are involved in emission or not. And if we look where volatile compounds are produced, in flowers they produce in inner epidermis cell layer and to a lower extent in outer epidermal cell layer for easy way to release to the atmosphere. In vegetative tissue, volatile compounds often produced in trichomes or glands, and they release from glands via mechanical disruption. And if trichomes or glands are not involved in formation volatile compounds, then volatile compounds produced in palisade mesophyll, in contrast to flowers where it's made in epidermal cell layer. And in this palisade mesophyll, it was found isoprene synthase expressed in palisade mesophyll cells. Of course, volatiles can be released out of leaves, for example, via stomata. But anyway, they really need to cross. Whatever they synthesize, VSEs have to cross cytoplasm, plasma membrane, and cell wall barriers to be emitted or stored in secretory structures. In general, volatile compounds, they are small, low molecular weight compounds, they are lipophilic, and they have high vapor pressure at ambient temperature. And for them, it's really very convenient and favorable, will be accumulated in membranes, but physicochemically unfavorable to diffuse out of membranes in the aqueous cell wall. And for many years, it was proposed that volatiles simply diffused out of the cells. And if they diffuse, this diffusion model, they lack membrane selectivity and directionality, and they will start to accumulate in any membranes within the cell. And it's very difficult to have control over cytotoxic effect of some volatiles, like aldehydes will react with free amino acid group of nucleic acid and proteins, for example. During my work over the years, what we found that not always internal pools of volatiles correlate with emission. For example, here you do see emission from snapdragon flowers over 12 days of lifespan of the flower. And below you have internal pools. And you can see that, for example, young flowers, they don't have accumulation internal pools inside, but they have high emission. And this is really very easy to imagine that volatiles which synthesize right away emits. However, if you look on old flowers, you can see that it's no emission, but volatiles start to accumulate internally. And if it will be simple diffusion, then it's practically impossible to keep these volatiles inside. It means that something prevents emission of these volatiles in old flowers. And this is what it says, that simple diffusion cannot explain observed volatile emission. And since we do know internal pools and emission rate, then we decided to make a calculation using fixed first law that if diffusion is the sole driving source for volatile emission, what should be actually flux? What amount should we have inside to sustain emission rate, which we do see? And here you can see emission rate, which we measure experimentally. This is concentration difference along the path. And here you have like resistance to diffusion across, and we have all these barriers shown here. And at steady state mass balance at each barrier, then rate of biosynthesis will be equal rate of emission. And using this calculation, what we found, we did calculation for two volatile compounds, methylbenzoate and nerolidol. This is phenylpropanoid compounds, and this is sesquiterpene compound, terpenoid compound. And what we found is that concentration of methylbenzoate in plasma membrane should be 120 millimolar, and nerolidol should be 48 millimolar. It's huge concentration, and accumulation of millimolar of VOC compounds in plasma membrane will be really cytotoxic, and it might cause major reorganization of cellular membranes and lead to leakage of organelle cellular content. And practically all these calculations showed us that biological mechanisms must exist to lower barrier resistance, thereby reducing VOC concentration in plasma membrane and likely internal membrane storm. And our hypothesis was that if emission is biologically mediated, it likely depends on secretory pathway vesicle trafficking, which already exists in a cell. And we started to look how these volatiles and what biological mechanism might involve in VOC emission. And practically, volatiles could be taken to plasma membrane in vesicles, or some lipid transfer protein shown here could take it to plasma membrane. Through plasma membrane, they can cross via exocytosis, or transporters can help to cross plasma membrane. And then lipid transfer protein is required in to cross cell wall and to prevent repetitioning of volatile compounds back to the plasma membrane. And then through the cuticle will be simple diffusion. And what we decided first to focus on, to focus first on looking for transporters across a plasma membrane. And our hypothesis was that biological players involved in VOC emission will be transcriptionally regulated with phenylalanine and volatile biosynthetic genes. Why with phenylalanine? Because petunia flowers produce predominantly Phe-derived compounds, and you can see here that the level of Phe is developmentally regulated with low level in buds and the highest on day 2 post-anthesis. And what we did, we generated RNA-seq data sets and at BAT stage and day 2, and we searched these RNA data sets for ABCG transporter with homology to ABC G11, Arabidopsis wax, ABC transporter as acquirer. And what we found several transporters with different level of expression, and one transporter was which exhibited 52% identity, 72% homology to ABCG11 Arabidopsis transporter, was upregulated more than 100-fold. And it was not only upregulated on day 1, on day 2 versus BAT, but it was also upregulated at 7 p.m. versus 7 a.m. It means, but if you remember, really expression emission is mainly occurs in petunia flowers during nighttime. And this transporter, when we search, we call this transporter ABCG1. It was already exists, this transporter, in data set, and it was written that it's transporter of unknown function, shown to be localized in plasma membrane, to the plasma membrane, and regulated by ADARANT1 transcription factor. And you can see here, this transporter was here, when ABC Arabidopsis transporter shown here. What we did, we down-regulated this transporter and generated 3 petunia lines with down regulation, and right away what we saw is that total emission was decreased and internal pools of these volatiles increased. And practically all compounds in petunia flowers were affected to different degrees. Then we overexpress this transporter in Nicotiana tobacco BY2 cells and perform assays where we incubated cells with methyl benzoate, radioactively labeled methyl benzoate, and we measured radioactivity. And depending on how a transporter is orientated in these cells, we could expect lower radioactivity in transgenic lines versus control, or higher radioactivity. And indeed, our lines had lower radioactivity. And this transporter could move methyl benzoate. We also checked benzyl alcohol, and benzyl alcohol was a substrate for this transporter, but this transporter was not able to move terpenoid compounds, menthol, and scleral analog. What is about toxicity? Our model predicted that volatiles will be toxic, and since we block movement of volatiles out of the cells, they should accumulate inside and lead to cell toxicity. And indeed, we use 2 different dyes to check for toxicity. One was propidium iodine, which stain nucleic acid only if membrane is damaged. And you can see here wild type, and this is transgenic plants. And second dye was fluorescent diacetate. And this practically dye is non-fluorescent, and it can get to the cells. But then diacetate moiety will be removed, and dye become fluorescent, but it cannot leave the cell if membrane is not damaged. And you can see that transgenic plants have less fluorescence because dye could leave the cell due to damage. We also mimic this phototoxic effect. We fed flowers with different concentration of benzyl aldehyde, and you can see at concentration of 15 millimolar, we can see already damage of the cells. And now it's our proposed working model for volatile emission, that volatiles will get to the vesicles will take them to the plasma membrane, then volatiles will be released to the cross plasma membrane, and then LTPs will take them across the cell wall, and then from cuticle they will be released. And cuticles, this is the final step from where volatiles is emitted. And when we check if cuticle accumulate volatiles, we found that approximately 52% of volatiles cuticle contain about 52% of volatiles, and the rest is accumulates internally. Then we decided to check what is the role of cuticle and what cuticle contribute to emission process. And since we also identified two ABCG transporters, ABCG12 and ABCG11 in petunia flowers, we decided to downregulate these transporters and to see effect on the cuticle and emission. We were not able to generate flowers for ABCG11, but we generated several transgenic lines for with RNAi downregulation of ABCG12 transporter. First of all, I want to say that both transporters are expressed in flowers, but ABCG12 has high expressions than ABCG11. And if you look at expression of these transporters over flower development, they express much earlier in flower development before you will see expression of synthetic genes. This is why we thought that it will be very good candidates for downregulation and changes of properties of cuticle. Here you have these transgenic lines which we generated, and we choose 3 lines with the most downregulation of expression of ABCG12 transporter, and you can see that wax amount was reduced up to 50% in this. But what was interesting that sorry, that practically ratios between constituents and composition of cuticle was not changed in this plant. When we check, when we look on the flowers will have some phenotypical changes. They were a little bit smaller and they're more translucent. And if when we stain it with toluene, you can see that control flowers will not stain with toluene, but transgenic flowers can be stained with toluene, showing that we have some changes in cuticle composition. When we measure the cuticle thickness was reduced by 22%, and also some cells, they lost their conical shape and they became flattened. And when we measure, it was about 23% of such cells in a flower. Translocation of VSCs across the cuticle involves 3 physiological processes. This is partitioning of VSCs from aqueous cell wall to the cuticle, diffusion through the cuticle, and then evaporation of VSCs from the cuticle surface to the atmosphere. And our model predicted that diffusion across the cuticle is a major barrier for VSC emission. Thus, our hypothesis was that thinner cuticle would permit increased VSC release. And when we measure total volatile emission, what we found that total volatile emission was decreased. Surprisingly, internal pools were also decreased, indicating that actually production of volatile compounds was decreased. And then we decided to calculate the total biosynthetic flux, which determine as a sum of total emission and changes in corresponding internal pools over time. And what we found that internal biosynthetic flux was reduced in these plants. What we wanted to know, at what stage of biosynthetic, this sand biosynthetic network changes occurs at the field biosynthesis up to here, at the level of substrate, or downstream at VIC biosynthesis. And to separate these two stages, what we did, we fed flowers with phenylalanine. Something is going on. With phenylalanine, and we were able to recover actually biosynthetic flux. And what it means that inhibition of VAC production occurs in the feed biosynthetic network, and the VAC biosynthetic capacity was not affected in these transgenic plants. But what was interesting that this feed feeding was not able to recover emission, indicating that practically like there is a mass transfer limitation in these plants. When we check genes involved in feed biosynthesis, we found that all of them were reduced. It was expression of Adarant1 was also reduced in transgenic plants, as well as expression of ABCG1 transporter, which transport volatiles out of the cells. These results practically show that cuticle serve as a central member of volatile metabolism. In order to count the reduced VAC biosynthesis and comparative analysis of wild type and transgenic plants, we define VAC emission factor, which is the ratio of emission flux to biosynthetic flux. And when VAF value is approaching 1, it indicates that emission is controlled mainly by biosynthesis. And when VAF is below 1 or less than 1, it means that it reflects the existence of mass transfer limitation. And if you can see here, in general, with cuticle, more should be emitted, or we should expect to increase in VAF. But in contrast, we found that VAF was decreased in transgenic flowers. When we look on individual compounds, we found that 2 compounds, methylbenzoate and benzaldehyde, they have VAF close to 1, indicating that practically their emission is controlled mainly by biosynthesis. But these 2 compounds, they have the higher vapor pressure and ambient temperature. And indeed, when we compare biosynthetic flux and emission flux, they overlay. But compounds with lower VAF, all the other compounds like benzyl benzoate and isoeugenol, ethanol, and benzyl alcohol, you can see that there is delay between biosynthetic flux and emission flux. And if you look in practically cuticle did not affect these volatiles with high volatility, but affected volatiles with relatively low volatility. If we look on effect of reduction in cuticle thickness on internal pools redistribution, what we found that it's less accumulates in a cuticle. And practically, you can see that cuticle is affected all volatile compounds. All volatile compounds were started to less accumulate in a cuticle. And practically, what is down regulation, it's led to this toxicity. And here you can see like a model how we envision what is going on. Here you have a wild type where volatiles and mainly here in cuticle and as they get into atmosphere. And here you have less volatile compounds in the cuticle. They mainly accumulate inside the cells. And cells start to sense it and downregulate production of phenylalanine and downregulate production of volatile compounds to prevent toxicity. And cuticle, as a result, cuticle serves as a resistant barrier for VOC emission. Cuticle holds about 50% of internal VOC pools, thus acting as a sink, storing VOCs to sustain efficient emission and protect cells from VOC toxicity. Cuticle provides little resistance for compounds with relatively high volatility, but does limit diffusion of VOCs with low volatility. The inability of VOCs to build up in a thinner cuticle leads to redistribution of internal VOC pools and results in cellular damage. Cells sense changes in intracellular VOC distribution and feedback inhibit VOC biosynthesis at transcriptional level. The decrease in cuticle thickness leads to a shift in mass transfer resistance to other internal barriers. And cuticle is more than a simple diffusion barrier, but is a member of VOC biosynthetic network. As you know, petunias produce mainly phenylpropanoid compounds, how I said it already. But, you know, Ben, when he came to my lab, he wanted to study terpenoid compounds. And he checked buds, flowers, closed flowers and day 2. And he found that they produced a little bit of terpenoid compounds. And out of these compounds, it was 4 saskaterpene compounds and 1 monoterpene compound, geraniol. And we were looking for genes responsible for formation of this. Found in our EST database 3 terpene syntheses, which is belong to TPS-A family, and 1 terpene synthase, which is belong to TPS-B family, and this is monoterpene synthase. And we overexpress this all 4 genes in yeast, and we found that TPS1 practically responsible for formation of almost all saskaterpenes found, and TPS4 and TPS3 produce only cadenine. TPS2 was geraniol synthase and is, as you can see here, it's expressed mainly in pistils, and geraniol also accumulates in pistils and correlates with expression of this gene over flower development, bud and flower development. The highest level out of all these saskaterpene compounds was in cadenine, because all 3 terpene syntheses produce this compound, TPS3, TPS4, and TPS2. However, and as you can see, cadenine accumulates in pistils, and it's also TPS3 and TPS4 expressed in pistils. However, TPS1, which is responsible for production of germacrine, bicyclic germacrine, and germacrine diol, is expressed in tube, but accumulation of volatile compounds occurs in pistils. What we did, we checked where it's in pistils, volatiles accumulate, and we found that they mainly accumulate in stigma. We also checked where expression of this TPS1 occurs, and it's not evenly distributed across the plants, across the tube, but it has the highest expression with close proximity to the stigma. And we also checked emission of volatile compounds, and we found that internal part of tube emits much more volatile, twice more volatile compounds than outside part of the tube. What we decided to check, if it's really tubes which produce these volatile compounds, we remove tube and continue to grow pistil, and right away we did not find these volatile compounds in pistil. these products of TPS1 products were missing. Second, what we did, we incubated tube in solution, which contains stable isotope labeled terpenoids, precursors. And we started to check if pistil or stigma, which never touched this solution, would accumulate labeled compounds. And you can see here the tube produce labeled compounds, and you can see that pistil also accumulates these labeled compounds. Next, we downregulated TPS1 expression, and right away you can see the tube stopped to produce these compounds, but you can see that they still produce cadenine, which is the product of TPS3 and TPS4. Right away, these compounds were missing in a pistil, but you can see normal production of geraniol, and also you can see some production of cadenine in pistil. Now, what we next did, we did complementation via gas phase. We started to grow a pistil within a tube from wild type, and you can see we were able to complement missing compounds. Why it accumulates in a stigma? Because stigma has the highest level of wax, and physicochemical properties this surface really determine why stigma traps these volatile compounds. And we also did exposure with exogenous caryophylline, and you can see again stigma trap more volatile compounds than the other parts. What we also found, and which was not really expected, is that these transgenic plants have smaller stigma, and you can see here that it was like both parameters, major axis, minor axis was reduced, pistil was, the weight of pistil was reduced, and diameter of style was reduced. But when we did when we started pistil within different tubes, and you can see here, when only you have wild type tubes, you can see normal growth of stigma. When you have a tube which does not produce terpenoid compounds, a stigma was always smaller. And what it means that practically pistil size phenotype is independent of pistil genotype, but depends on the tube genotype. Also, when we see fumigation by tube does affect seed yield, and you can see that transgenic plants produce less seeds, but the seed mass was exactly the same, but amount of seeds per flower was reduced. It was fumigation by tube does not affect pollen movement, which was exactly the same in wild type and transgenic plants. But what we know, what is not clear, if it's effect of community, microbial community, and what we did, we took petals from transgenic plants and wild type tubes, we incubated them in a liquid and then plated overnight, and you can see that practically presence of terpenoid compounds drastically reduced microbial community on a pistil. And we also checked OTUs, operational taxonomic units, and if you can see here on day 0, practically no difference between wild type and transgenic plants, but on day 2, they accumulate much more OTUs, and it's mainly Pseudomonas. It means that terpenoids play a role in shaping bacterial community of petunia reproductive organs. And what we wanted to know is it small size of stigma is only because maybe bacteria present like microbiome, or it's really effect of terpenoid compounds. And what we did, we actually bleach stigma, and you can see here after bleaching, practically it's no bacterial growth, and then we did a complementation, and it's really independent of was it treated stigma or not treated with bleach, you can see the same effect of terpenoid compounds on growth of stigma, indicating that practically microbial community does not affect stigma development. And here, in a summary, natural fumigation enables directional transport of volatiles from one organ to another. And during flower development, you can watch here, TPS is expressed in a tube, and volatiles are emitted inside the buds, and they accumulate in reproductive organs. And since terpenoids are known to defend against florivore insects and microorganisms, then they protect reproductive organs. And when flower opens, reproductive organs are protected to ensure successful pollination. And terpenoids are required for stigma development and seed yields, and transmission of terpenoids from tube could serve as a mechanism to coordinate the timing of pistil maturation and petal development. And I want to thank all people from my lab, my collaborators at Purdue University, in Belgium, Amsterdam, and Austria for their contribution to this work. Mainly, work has been done on cuticle by pen layer, on ABC G1 transporter by Fumi, and the work was supported by National Science Foundation, USA, and NIH, and all these agencies. And thank you very much. I want to finish here and answer your question. And what I want to say is that new book, Biology of Plant Volatiles, just came out. Thank you very much.
Dr. Manoj?
Yes, sir. You can start.
Okay. Okay. Thank you, Natalia, Professor Natalia, for giving such a wonderful talk. It was very interesting and very insightful, especially on volatiles, their formation, how they are like biosynthesized in plants, and how their biosynthesis is regulated, and finally for giving the newer insights beyond the traditional one, like how the volatiles are released to the environment. I'm sure all the attendees would have enjoyed the talk and got some newer insights, and to to listen from your talk, and to listen from an authority like you on volatiles. It is a great pleasure for all of us. I'm sure all the attendees would have benefited from your talk. Thank you very much again for joining the session at the late night. Almost, I think it's a midnight for you. So thank you very much. So can I now request our director, Dr. Prabodh Trivedi, to release the e-souvenir?
I guess Professor Mande DG, if he is there, maybe we can release the e-abstract book and souvenir. Dr. Manoj?
Sir, he is not there, I think.
Okay, so maybe you can just bring that on the screen and we can release the e-abstract book. Yes, Dr. Manoj, you can click it.
Sir, it has been Right on the screen, sir.
Okay.
Manoj,
Yes, sir.
what about the abstract book?
We have released it, sir. It has been displayed, sir.
Okay, it got displayed. Okay,
Yes.
good. So now maybe Dr. Sasne will give the vote of thanks.
Thank you, Dr. Prabhu. I'm privileged that you have given me the responsibility to give a formal vote of thanks. We are grateful and really indebted to both the dignitaries. First of all, our Director General and the great leader of CSIR and Secretary Department of Science and Industrial Research to be here, giving his valuable time to be with us to inaugurate this function, this seminar, and we are really indebted to him. Both are great leader in their own science. And Natalia, really, you have found out your time, and in the midnight you have delivered the lecture for the benefit of people around the world. The scientists, researchers, students, they are all benefited. So we are grateful to you, and really, frankly speaking, we are proud to be a part of your lab, myself and Dinesh, together. And coming to our director, Dr. Prabhat, he is always supportive and always encouraging us to do such things in a bigger way. And Dinesh, he has organized, and thankful to Dinesh also. Director CIMAP has given the full freedom to Dinesh and the organizing committee to work on this and for organization, and really thankful to director and Dinesh and all the organizing committee from Bangalore as well as from CSIR-CIMAP, the IT team who are organizing, all students who are participating in this, research students who are participating in this particular seminar, gaining lots of insight. And the first lecture is the mirror of the seminar, and what a great lecture it was. And coming to all the scientists and my CIMAP colleagues and all the speakers around the world, we thank all of them on behalf of CSIR-CIMAP and Director CIMAP that they are here listening to the lectures and giving and interacting with each other, and press, media, our finance, administration. I thank everyone to help us to organize this seminar. And Abha, thanks for comparing. And if I have forgotten anyone, please kindly forgive me. Thanks everyone to be with us and in the great inauguration and great function. That's the seminar, online seminar happening in CSIR-CIMAP. Thank you very much.
Abba, I think Professor Natalia has already left. So
Yes,
we will
she has left.
we are on time, and we will move to the next session that will be on biosynthetic pathways of secondary metabolites.
So we should get out of this and enter into the new link, or what?
We have to.
Actually, sir, the session has already started by Dr. Venkat. So we have to leave this session and join the session.
Okay.
Okay.
Thank you.